Thanks for having me. This has been a really awesome conference. I've been quite stimulated, and now we'll talk about simulating the brain and the body. These are two separate projects that I'll talk about, and our dream is to bring these two together. I want to start by acknowledging the people who did the work for the two projects. At the top is my close collaborator Jakob Make and his student Jana Lappalainen, along with a bunch of my colleagues at Janelia who did the first project, and the second project was a big collaboration between many colleagues at Janelia as well as folks at Google DeepMind. Okay, so you've heard about the fly a lot over the course of this meeting, so I won't talk too much about the fly itself, but I want to introduce one of the stars that, something that happened just 2 weeks ago was my colleagues at Janelia released a connectome for the entire nervous system of a male fruit fly. And this is that connectome. Each color here is a different neuron, and so we can see the shapes of every neuron, as well as which other neurons they're connected to. So it's a wiring diagram for the entire connectome. In my lab, I did my PhD working on machine learning methods to create these connectomes. And now that we have them, we're working on trying to figure out how to simulate them, how to extract understanding from these wiring diagrams. I'll give you my caricature as a computational neuroscientist of how the brain works. So at the bottom, at the microscopic scale, are the cells and the molecules that make up the brain. The brain is made out of brain cells. We care about how they're connected. That's the connectome. We also need to know the biophysical properties of neurons and synapses. So what are the input-output functions of individual neurons and the synapses that connect them? How do they work? And then there's a host of other mechanisms at the microscopic scale that include neuromodulation, plasticity, synaptic plasticity, and more things like that Misha, for instance, talked about, and things that we hope to discover soon. Have yet to discover. Collectively, this microscopic scale collectively gives rise to neural activity of the brain. And the neural activity collectively across the brain gives rise to the behavior that we observe. Now, if we can measure everything at the bottom scale perfectly, then we could imagine building a simulation that perfectly predicts neural activity across the whole brain and then predicts the behavior across the whole brain. Unfortunately, we can't yet do that. The new exciting thing that we now have is the connectome, and we have it quite comprehensively, as I showed you. But we cannot yet comprehensively measure the biophysics of neurons and synapses. The neural connectivity we can get from a dead brain. That's what makes it easy. Excuse me. These biophysical properties have to be measured in a living brain, and so that's a challenge right now. And so because of this, when I was doing my PhD about 15, 20 years ago, there was a big debate about the utility of connectomes. Connectomes are going to be really expensive to get, take an enormous amount of work. Literally, people were debating whether or not we should do connectome projects. One of the arguments, which is correct, is that if you can measure only the connectivity, but you don't have all of this, then really is that enough because you only have an incomplete view of the microscopic scale. So the talk I'll tell you about is trying to figure out how to get past this limitation of not having direct measurements at the microscopic scale. So the good news is that we've been able to record from other scales, the more macroscopic scales, for quite some time. We've been recording behavior, animal behavior with their eyes and then with cameras for a very long time. We can record neural activity, not everywhere, not all the time, but in certain conditions you can record neural activity. And so I'm kind of color coding it a little bit lighter. And we know something about the space of possible single neuron biophysics and synaptic biophysics. We may not know the exact details of every single neuron, but we know something. So, the thought that we had was, can we use machine learning methods to take measurements at the macroscopic scale and then use them to inform the parameters at the microscopic scale? And if we can do that, then we can do this sort of take measurements from across the different scales, the microscopic and macroscopic scales, to put together an integrated model that goes all the way from single cells and in some cases molecules, all the way up to behavior. So that's the dream. And let me tell you then about the first sort of attempt we made of doing this. And so here the question we asked was, let's look at the very extreme case of only having the connectivity and only knowing at like the highest scale, the behavior or the computation performed by a neural circuit. Let's say we know that just that. How far can we get? And this is a way of sort of characterizing really, how strongly does the connectome alone constrain any computational model that we can build? Okay, so exactly what we did was we took connectomic measurements from the motion pathways of the fly visual system, and then we built our simulation. The simulation is built without ever using any neural activity measurements, but it can predict neural activity measurements. So now we can ask, how well does our model predict neural activity measurements, for instance, those that have been made in the lab across the motion pathways across various cell types here. And then we can compare those measurements made in the lab to the predictions from our model and see how well we're doing. How well does this model work? Okay, so a quick introduction to the fly visual system. The fly is highly visual. It has two large compound eyes that dominate its head. And you can see that the pixels of the eye, the ommatidia, are organized in a perfect crystalline hexagonal lattice. That's actually really how it looks. And the cool thing is that the neural architecture, the neural network that processes this visual input, also has a perfectly crystalline hexagonal lattice. There's the retina, the pixels, the photoreceptors are organized in that way. And then there's the lamina, medulla, lobule, lobule plate. These are called columns, and these columns are arranged in a really very repetitive pattern. So we have, at this point in time, we didn't have the full connectome for the optic lobes. In fact, what we had was across a variety of different studies, connectomes for various pieces of this. So we curated all that data across many studies to put together a consolidated connectome for the motion pathways of the fly visual system. So this is 64 cell types. This tells you which cell types are connected to which other cell types. And so you see the photoreceptors over here, Rs1 through 8, and then there's a whole host of other things. I'll remind you that the fly has such a stereotypic nervous system across individuals that there's names for every neuron. And so these are the names, and don't worry if they don't make any sense to you. They're just letters and numbers, but I'll try to interpret them. So first thing to notice is that we have this connectome. It's sparsely connected. So most of the cell types are at the type level are not connected to each other. Second thing to notice is that there's these squares. They have different sizes, and then they also have different colors. We curated a data set also of gene expression data across these different cell types to make predictions for whether a cell type was an excitatory cell type or an inhibitory cell type based on the neurotransmitters that they make, as well as the receptors that are expressed on the postsynaptic side. So for each of these connections, we know whether it's excitatory or inhibitory. And then the larger squares tell you how many synapses a pair of neurons of cell type A and cell type B have. You'll note that there's a very large dynamic range here. There's some pairs that have over 100 synapses between neuron 1 and neuron 2, and others that may have a handful. So this has actually been shown in the fly to be a good proxy for synaptic strength. This will be important. We also know the spatial pattern of the connectivity between cell types. Here's 2 different cell types. If you know about convolutional neural networks, you can now start thinking about these cell types as feature maps, and then the connectivity pattern between them as convolution filters, so to speak. And so what we built is basically a hexagonal lattice convolutional network, recurrent hexagonal lattice convolutional network model of this fly visual system. So you have the retina. So each hexagon here is all the neurons of a particular cell type, and each pixel inside of that hexagon is one of those neurons. And our job is, let's say we show a video clip to the retina. Our job is to try to predict the neural activity as it propagates through the network. So what would be the gray level, the activity of any of these neurons? So that's what we're trying to do here. So if we want to, the connectome is static, comes from a dead brain. So in order to simulate it, we need dynamics. So let me tell you about how we did that. For this study, what we wanted to do was to say, let's take the detailed connectome, but really simplify everything about the biophysics of neurons. We know the shapes of these neurons in great detail. We could build, highly detailed compartmental models and, do a lot. But instead, here, it's quite simple. And so even the simplest model has some parameters that we can't measure directly. And so here's the simplest model that you could imagine. We're modeling the voltage dynamics of each neuron. And there's 2 parameters to it. One is a time constant of a neuron, which tells you how quickly does it respond to inputs. And the second is the resting membrane potential, which says, in the absence of any input, what is the voltage that it sits at? So that part is called the passive point neuron. These neurons in the early part of the visual system don't spike. So it's actually a good approximation for how the system works. And now, this is the cool part. We're going to use the weights in this model are going to come directly from the connectome. And so the synapse count, so this box over here is directly measured. This is a combination of the transcriptome and the connectome. So Nij is the number of synapses between neuron I and neuron J. Sigma ij, T is a type of the neuron, so Ti, Tj, just tells you whether it's a excitatory neuron plus or minus, the sign of that connection. So that's measured. There's a thing that we don't really know. We don't know if there's 10 synapses between a pair of neurons, how strong is one of them? How much current flows through one of them? So that's a free parameter. That's a synaptic biophysical parameter, even in the simplest model. And we call it the unitary synaptic strength. So in this model, there's 3 classes of biophysical parameters, 2 that are neuronal, the time constant and the resting membrane potential, and then the third that's synaptic, that's the unitary synaptic. So what we said is, we largely know the connectome should give us. Share these biophysical parameters according to cell type for the neurons and according to synapse type for the synapses. And in this case, we say that any pair of cell types has its own unitary synaptic strength. And so that's why they're indexed by Ti and Tj. So the cool thing is, now this model, instead of having millions, actually, it should be about 2 million, at least, free parameters, which would be the case if we had trained a deep neural network in this way and had to estimate the weights. Here, because we know the weights and instead don't know the biophysics, we only have 734 free parameters because we share them by cell type. So it's a massive reduction in number of parameters. awesome, but it's still a lot. So how do we set them? This is where we took inspiration from the task optimization, the neuro AI procedure for building models using task optimization. We said whatever those biophysical parameters are, they should be good for doing vision. This network somehow needs to do vision. And in particular, because these are the motion pathways, they need to do motion vision. So let's see if we can train this model. In this case, we're not training the weights, we're just training the transfer functions, basically, of each neuron and synapse, in order to perform an optic flow prediction task. So this is a classic computer vision task. So there's video clips over here. The video clips are rendered onto the fly eye and passed into the retina. We're now integrating there's 45,000 neurons here. We're integrating the couple differential equations of those 45,000 neurons at tiny 20 millisecond time steps. And we're asking that the feature representation that's computed by this network at the second half of the network form somewhere a representation of optic flow that we then, using a motion decoder, transform into the coordinate system that computer vision people use, which is a vector field that looks like this, which for each frame tells you where does that pixel move to in the next frame. So for each pixel, there's an arrow that tells you where that pixel moves to in the next frame. So that's what we try to predict. So we do backpropagation through time. This is a little bit of a harder machine learning optimization problem because this network wasn't trained, it wasn't designed to be trained. And in fact, usually you fix the input-output transfer functions, right? The nonlinearities, and train the weights. Here, we're fixing the weights and training the nonlinearities, and so it's a bit trickier. But we figured out how to solve those problems, and so now. We have a simulation that we can then show video clips to, including the kinds of video clips that people showed and have been showing in the lab. The motion pathways are the most studied part of the fly visual system for a few decades. And so we can ask each of these cell types that have been characterized in the lab, what do they look like in our model? And so we curated now, again, a data set of about 26 studies that have characterized neurons in the motion pathway, and then use that to generate stimuli that we then showed our visual system and asked, what do the neural activity predictions look like? So one of the most important discoveries, I think, in the fly visual neuroscience was the discovery that these two cell types, which have the exciting names of T5 and T4, are the elementary motion detectors. So that means that the input neurons to these cell types are not motion selective. These neurons are motion selective. So they compute motion from their inputs. And what that means is that if you take an edge stimulus that looks like this, and then you move it around in 8 different directions from the center out, and you record its neural activity pattern, it will fire more strongly for motion in one direction than motion in the other directions. You can characterize that with the polar plot that looks like this. This is the seminal study that first described these. So in white, you can see for each of these cell types and these 2 different stimulus types what the actual tuning should look like. And in color are our predictions. And we were super excited to find that now, using this procedure, we could predict, if we were to time travel back to a world where we had the connectome first and hadn't been recording from each of these neurons, we could then predict which neurons would be the motion detectors and do it quite nicely. And you can even characterize across these 64 cell types, which are the neurons that should be motion selective, and it should be these 2. It should not be their inputs, and they're not. And then there's some novel predictions over here, Now, another class of sort of characterization that's been performed is most sensory systems segregate into on and off channels. These pathways in the visual system I mean, only half of these cell types have been experimentally characterized to date. And so for the ones that have been characterized in these 26 studies, we asked, which of these are known to be on and which are known to be part of the off pathway, which means that they respond more strongly to decreases of light intensity versus increases of light intensity, and we got them basically right for all of them. So what does this plot look like? Here is what we call the flash response index. Does it respond more strongly to increments or decrements of light? And each of these circles is a different model. So what we did was we didn't just train one model. We trained 50 models. And we asked, do these models come up with the same solution or different solutions? So each of those circles is a different model. And while it's not like a single model gets all of these right. If you look across this ensemble of models, you find that the median actually is a very good predictor. And so this ensemble is something that we think is very important way of using these connectome-constrained models, as we call it. They generate hypotheses the way that we are using them, and the ensemble characterizes the space of hypotheses that are consistent with the task as well as the connectome. And so we've done some work, hypothetical optimal experimental design with this, showing that you can use this ensemble, this solution space, and explore it and figure out which neurons you should record from to optimally constrain the rest of the network, which is very exciting. Now, the actual prediction is really a time series. In response to a video clip, there's a time series of voltage in one of these neurons. So this is the voltage. It's the prediction of what the voltage is in response to stimulus in one direction versus the other. But in actuality, we also have underlying it the currents. We can say what are the excitatory and inhibitory currents. People have recorded voltage, but it's harder to directly record these currents. You can make inferences about them, and harder still is to make predictions about the precise currents that come in from different cell types in order to give rise to direction selectivity. So this mechanistic simulation can now allow us to sort of dissect these computations and figure out the mechanism underlying computations in a nice way that you can't experimentally do. So that was a quick summary of the work. We're continuing this work. We first worked on this motion pathway. That's what I told you about. Since then, this year, there's now publication of a connectome for the entire optic lobe, not just a small piece. And so has a lot more cell types and a few more neurons. And so we've now begun working with this connectome, and actually the new connectome combined with more detailed biophysics actually is not only easier to optimize, we were very excited to find, but also makes more reliably the correct predictions. Whereas in the old models, there was more variability. Here, there's really crisp sort of predictions. So this is teaching us that getting the connectome right, getting all the details of the connectome really helps, but also getting the biophysics right helps. So we're gradually increasing the biophysical complexity, and it seems like that helps. We're also integrating transcriptomes, looking at the transcriptome of the receptors, the precise receptor subtypes that are used, and then grouping synapses into types based on that. And with the transcriptome, that actually improves performance. And the cool thing is, it improves performance even for neurons that were not task constrained. So here is a neuron that's not task constrained, but because it shares the same parameters, biophysical parameters, that you can link them through the transcriptome, you can now predict their properties, which is a very exciting way of bringing together these measurements that we have of the connectome and the transcriptome to actually make good ground this model. This was possible only because we have this connectome. If we had a black box neural network and we trained it to perform the same task, we would not be able to have this degree of prediction. In fact, the hidden units of this network would not be identifiable. And it's only with the connectome that they're now identifiable and you can predict their properties. So, we were excited by this approach and excited by the fact that we have more connectome data. What we need for our approach is a characterization of the input and outputs of a particular circuit, so the computation that's performed. So if we want to scale this up to the whole brain, we need to know all of its inputs and all of its outputs. And so for this, the inputs of the brain are, of course, the body, and the body is what mediates between the environment and the brain. And so we now need a simulation of the environment and the body. And so this is, in the last 3 minutes, I'll tell you about that work. And I'm very pleased to precede because our esteemed speaker who's coming next actually did very good work on building their own biomechanical body models of the fruit fly that preceded our work. And of course, there's speakers from this work as well here. So what we did, my colleague Igor Sivanowitz, who's one of the world's best insect anatomists, imaged an entire fly and then built a Blender simulation, a Blender model, 3D model of the body, which then we collaborated with Yuval Tassa and Josh Merrill at Google DeepMind to bring into the MuJoCo physics simulator, and then gave it some extra, added some physics to the physics simulator to make it walk and fly. And then we did reinforcement learning, and ta-da, it works really well. It can walk, and it can fly. But it does it nothing like the way the real fly walks and flies. So that was a big disappointment. We did all this work, and we thought it would just work right away. And our colleagues were saying, just work harder. Those are the biologists. This has to work. What we did instead was we worked with our colleague Kristen Branson and Alice Roby, who is very good at recording fly behavior, the way that real flies walk. And then we used that to sort of build a training set of how real flies walk and fly, also using data from Michael Dickinson's lab. And then once we used a form of imitation learning using reinforcement learning, we were then able to get the fly to walk better and more realistically by training it to match the way that real flies walk and fly. Now, that was not the end. That was the end of that paper, but now we're still working away to add more realism to this body. So this is very much work in progress, but this depiction here tells you all the muscles that we're working on rigging, the same dream team, Igor and Roman, in collaboration with Bing's lab and John's lab, including Elliot, who's here, have been working on putting this together. And so I hope we'll get to report on this imminently. And so the dream, my dream, is that we can now use this collection of data that we have, this multi-omic data, all the way from molecules to behavior, to put together a foundation model, not the kinds of foundation models we've talked about so far, a mechanistic foundation model that matches on a one-to-one basis the neurons and the connections that we get from the connectome, the biophysics as dictated by transcriptome, as well as behavior and neural activity. The one challenge is that these data sets are available in comprehensive form. You can download them off of web servers. There is no big data set no comprehensive data set. This is what Eva was talking about as well, of neural activity and behavior. And so we need to start curating it if we want to make this an AI-driven modeling approach like this to work. And so I'd like to start working on this, and I would be very happy to collaborate with folks here to put together a data set like that. Before I end, I want to say we're very excited. This is, I think, a new phase of computational neuroscience, where we now have these new data, and we now have to figure out how to use that data to build new computational neuroscience models. And so we're running a winter school, and there's still a week left to apply. We have a really fantastic set of lecturers. It's 2 weeks long, and it's basically the computational neuroscience of connectomics, the new computational neuroscience. We hope you'll apply. That's the website. And with that, I'll thank you. And there may not be time for questions, but thank you.
Thank you.